Biochemistry. Life and living systems appear to represent negative entropy. Unlike most of the observed universe, they grow more structured and more complex with time. This concept and the term negative entropy were perhaps first introduced by E. Schrödinger in his 1944 book What is Life? The concept has been widely debated, and others have proposed several terms, such as egetropy and cytropy, as alternative nomenclature. The fact remains, however, that among systems subject to the laws of thermodynamics, life displays the unique properties of increasing complexity, self-regulation, reproduction, and before death and decay, negative entropy. One could argue that life displays this unique property for a finite time and then yields to the normal laws of thermodynamics and adds back to the general randomness and entropy of the universe. At the cellular level, a highly complex, redundant, and highly regulated macromolecular and molecular set of machinery maintains this negative entropy. At the tissue and organism level, this level of regulation includes internal, external, and cross-species forms of communication and regulation, at least in the gastrointestinal tract. This chapter reviews the biomolecules, macromolecules, and macromolecular systems that are responsible for this machinery, regulation, and communication. The major groups of biomolecules are amino acids, fatty acids, carbohydrates, and nucleic acids. These building blocks, in turn, form the major macromolecules, proteins and enzymes, lipids, glucose and starch, ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Other biomolecules, such as coenzymes, vitamins, are briefly reviewed. In addition, enzymes, the subset of proteins that actually forms the cellular machinery, are highlighted. Enzymes have a primary role as catalysts of the myriad reactions necessary to carry out cellular development, cell metabolism, growth and differentiation, and cellular regulation, and they ultimately control cell death, apoptosis. The critical metabolic pathways of energy production from sugars and fatty acids are reviewed. Nucleic acid metabolism, a critical area for cancer pharmacotherapy, is reviewed, including DNA replication and repair, RNA production, control, degradation, and structural roles, and protein synthesis and post-translational modification. The area of recombinant DNA technology is introduced solely with a nod to therapeutic protein production. 5:1. Chemistry of biomacromolecules, proteins, carbohydrates, fatty acids and lipids, and DNA and RNA. Proteins. Amino acids are the building blocks of proteins. Table 5:1 lists the common protein-incorporated amino acids by name, indicates their side chain structure, provides their 3-letter and 1-letter abbreviations, and summarizes their chemical characteristics, malmi, hydroxyl, and so on. Although all amino acids contain an amino group and a carboxylic acid group, they fall into 2 large groupings: primary and secondary amines, depending on the structure of the amine group. The nitrogen atom in primary amines has only 1 bond to a carbon atom, whereas for secondary amines, the nitrogen atom is bonded to 2 carbon atoms. Table 5:1. Common protein-incorporated amino acids. As the precursors for synthesizing new proteins, the amino acids are clearly important from a nutritional perspective. The 9 amino acids essential in human nutrition are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. They cannot be synthesized from other amino acids or dietary constituents. Adults can produce tyrosine from phenylalanine and cysteine from methionine. However, infants and young children have only a limited ability to make these conversions. Hence, tyrosine and cysteine are considered conditionally essential in this population. The remaining 9 amino acids are considered non-essential, indicating that they can be synthesized from other amino acids or metabolic intermediates. Many amino acids are involved in critical metabolic pathways. Arginine, ornithine, and citrulline are the 3 amino acids of the urea cycle, conversion of ammonia to urea, which is the main pathway for excretion of nitrogen waste product from protein metabolism. The urea cycle is coordinated in the cytoplasm and in the mitochondria of the cell. Another example is the tripeptide glutathione. It is a first-line cellular antioxidant formed from the amino acids glutamine, cysteine, and glycine. It is synthesized in all tissues by a 2-step enzyme-mediated process.
Proteins are made from long chains of amino acids. Amino acids all have a similar structure, with carboxyl and amino functional groups. What makes each amino acid unique is the presence of a variable, or R group. The R group gives each amino acid its chemical properties. Amino acids are linked together by peptide bonds, forming long polypeptide chains. As these chains form, the polypeptide develops multiple levels of structure, which contribute to its overall shape. These levels are called the primary, secondary, tertiary, and quaternary structures. The primary structure is the order of amino acids in the polypeptide chain. Amino acids can be likened to letters in the alphabet. Just as thousands of words can be constructed by varying the 26 letters in our alphabet, so too can different proteins be created by varying the number and sequence of just 20 amino acids. The secondary structure refers to the pleated sheet or helix that a protein chain can form due to hydrogen bonding and other chemical attractions between the R groups of nearby amino acids. The overall shape of a polypeptide is called its tertiary structure. This level of structure is created when the secondary structure folds and twists upon itself. The tertiary structure is held in place by a variety of bonds and interactions that form between the R groups of the amino acids in the chain. Once polypeptides have formed a tertiary structure, they are commonly referred to as proteins. In some cases, proteins can form quaternary structures, which consist of interactions between multiple proteins. These interactions often result in the formation of large protein complexes. Examples of proteins that have quaternary structure are hemoglobin and the sodium channels in the cell membranes.
A peptide bond formed between 2 amino acids is the primary element of peptide and protein structure. The formation of this bond is orchestrated at the cellular level by a very complex structure called the ribosome and involves several forms of RNA. However, the net chemistry of bond formation and hydrolysis can be easily depicted as. Note that in the bond formation direction, a molecule of water is eliminated, whereas in the reverse, bond hydrolysis direction, a molecule of water is inserted. Protein and enzyme structure has 4 main elements: primary, secondary, tertiary, and quaternary. Primary structure is the linear sequence of amino acids. Secondary structure is the first step in folding up the protein and begins with local groups of amino acids forming into 2 main structures. Alpha helices are formed from predominantly hydrophilic amino acids, and beta-pleated sheets are largely formed from hydrophobic amino acids. These 2 sets of structures form largely through hydrogen bonding. Tertiary structure is the more complex folding and arrangement of these helices and sheets into a globular structure, which is held together through hydrogen bonds, ionic interactions, and disulfide bridges. Disulfide bonds provide some thermodynamic stability, but they mainly aid in proper protein folding. Tertiary structure may also incorporate non-amino acid components, such as metal ions, as a structural element. These elements are usually bonded to functional side chain groups from amino acids in disparate parts of the primary sequence of amino acids. Thus, they can be structural elements or functional elements providing additional chemistry, in the case of enzymes and transport proteins. Quaternary structure is the combination of two or more individual protein chains to form a multisubate protein complex. This structure is formed and maintained by hydrogen bonding, as well as by ionic and hydrophobic interactions. Quaternary structure of proteins allows greater regulation of transport function, i.e., hemoglobin and oxygen delivery, and enzyme activity, i.e., cooperativity and on-off control. Figure 5:1 depicts these four elements of protein structure. Mutational changes in DNA can lead to abnormal proteins. Thus, a single nucleotide polymorphism, SNP, in the sequence of the coding DNA can lead to an alteration in the primary amino acid sequence of the resulting protein. Sickle cell anemia is a good example. Normal adult hemoglobin has a glutamic acid at the 6th position in the beta-globin protein chain. Because of a single point mutation, AT, sickle cell patients have a valley at this position. This change in beta-globin structure leads to change in the overall hemoglobin, 2 alpha-globin chains and 2 beta-globin chains, structure, which leads to a sickle shape in the red blood cell and occasional blockage in the microvasculature, known as sickle cell crisis. Thus, a single change in a nucleotide in the DNA coding sequence can lead to a dramatic clinical effect. In addition, many proteins undergo further modification after proper folding and assembly. These changes are termed post-translational modifications. Table 5:2 lists the most common of these modifications. Although amino acids are the building blocks of protein, sugar molecules and fatty acids are the building blocks for other essential cellular components. Carbohydrates. Carbohydrate is hydrated carbon, with the general chemical structure of CH20. The most generally used structure is C6H12O6. Indeed, the most common carbohydrates are hexoses and ketoses, C5H10O5. Carbohydrates are important elements of cellular energy as well as important components of cellular structure. Table 5:3 lists some of the most common carbohydrates as well as their occurrence, gross structure, and uses. From a nutrition perspective, carbohydrates are a major source of energy. In humans, carbohydrate energy is stored as glucose, the animal form of starch, predominantly in the liver and muscle. During fasting, glucose can be hydrolyzed to release glucose units to maintain blood sugar levels at a minimum level. This process can easily surpass an overnight period of not eating, i.e., fasting for 8 to 12 hours. Ketoses are components of nucleotides and thus are part of RNA and DNA. Note, MW equals molecular weight. Sugar molecules are also important in glycosylation, the post-translational modification of proteins. The specific scheme of glycosylation of cell surface proteins is essential to cell-cell recognition. A good example is the ABO-human blood groups. Specific glycosylation of cell surface proteins, glycoproteins, as well as the same specific glycosylation of membrane lipids, is the base of the human blood groups. Figure 5:2 shows the specific groupings of carbohydrates that delineate the most common ABO-human blood groups.
Most proteins and many lipids of the cell membrane are actually glycoproteins and glycolipids to which short oligosaccharide chains can be added. For example, there is an oligosaccharide chain which determines blood type, which can be added to proteins and even some lipids of red blood cell membranes. In this chain, the first sugar added to, say, the protein is N-acetylgalactosamine. The second is galactose. The third is N-acetylglucosamine. The fourth is galactose. And the fifth, known as fucose, is added by the enzyme FUT1, or galactoside 2-alpha L-fucosyl transferase. This is an important reaction because once this is done, this protein and sugar chain is considered to be the H antigen. If someone has blood type O, this is the end of the process, and this oligosaccharide chain is what blood type O individuals possess. However, another enzyme, known as glycosyl transferase, may attach another monosaccharide group to the end of this chain, depending on which allele of the enzyme an individual possesses. Type O individuals possess mutant alleles which do not function, so no more sugar groups are added. If someone possesses the A allele of glycosyl transferase, then the sugar N-acetylgalactosamine is added. And this is the carbohydrate chain which defines an individual as blood type A. If an individual possesses a different allele of the enzyme glycosyl transferase, then the monosaccharide galactose is added at the end of this chain instead of N-acetylgalactosamine. And this oligosaccharide chain will define individuals with the blood type B. Individuals who are blood type AB will possess two different chains, one which ends in galactose, this is the B oligosaccharide, and one which ends in N-acetylgalactosamine, this is the A oligosaccharide.
The glycosylatio pattern shown is for red blood cell membrane proteins glycosylated at the THR or SO residues of specific membrane surface proteins. Glycosylatio of membrane lipids also occurs. Note that two genes control the addition of the last carbohydrate. Type A people express two copies of the gene that codes for A pattern glycosylatio. Type B people express two copies of the gene that codes for B pattern glycosylatio. AB people express one copy of A and one of B. Type 0 people express neither A nor B pattern genes. Fatty acids and lipids. Long-chain fatty acids, as components of phosphoglycerides, are necessary for the structure of cell membranes. They are also the precursors for molecules of cellular communication, the prostaglandins and the leukotrienes. In addition, they can be stored for a long period and later converted to cellular energy. Lipids are fat-soluble, lipophilic molecules and include fatty acids, cholesterol, some fat-soluble vitamins, and glycerides. Fatty acids not only are a major source of energy but also are critical components of cellular membranes as phospholipids. The glycerides are combinations of fatty acids esterified to a glyceryl backbone. Phospholipids are glycerides in which a phosphate group, plus other molecules, is also linked to the glyceryl backbone. Figure 5-3 depicts the structural elements of the most common phospholipids. Note that a proton is lost from each X molecule in the formation of the bond with phosphate. Table 5-4 lists the major fatty acids incorporated into phospholipids. Amuglyceride has a single long-chain fatty acid attached by an ester linkage to a single hydroxyl group on glyceryl. A diglyceride has two long-chain fatty acids attached to glyceryl. A triglyceride has all three hydroxyl positions on glyceryl esterified to a fatty acid. For most phospholipids, the two fatty acid chains differ from each other. DNA and RNA. The information to build a cell, tissue, organ, or living organism is contained in the genetic code. Construction, maintenance, and transcription of the code, DNA to messenger RNA, as well as translation, messenger RNA to protein, involves polynucleotides, DNA and RNA. The nucleotide units of both DNA and RNA are composed of three basic components: a purine or pyrimidine base molecule, a sugar molecule, D-ribose in RNA or D-2-deoxyribose in DNA, a phosphoric acid molecule. Figure 5-4 shows the chemical structure of the DNA and RNA nucleotide bases. DNA has the single primary role of storing the information, genetic code, of the cell. RNA has several varieties and unique structures that perform specific functions in transcription, translation, and control of both transcription and translation. Following is a summary of the major groups of RNA and their functions. Messenger RNA, mRNA, is a processed transcription, or copy, of the DNA. Particular parts of the DNA, intros, have been eliminated, and the fused structure contains the sequence, composed of the sequences in the exos, of triplet bases, codons, to code for each amino acid in the linear sequence of amino acids that form the primary structure of a protein. See Chapter 6 for additional details of mRNA synthesis. Transfer RNA, tRNA, is the highly ordered sequence of nucleotides folded into a cloverleaf-like, inverted L structure with internal. Note: thymine, DNA, and uracil, RNA, differ only in a methyl group. Thus, each amino acid incorporated into a protein linear sequence has a unique tRNA. Ribosomal RNA, rRNA, refers to the two large RNA molecules, 1 to 2x 106 molecular weight, that bind a multitude of proteins to form the two subunit, 30S and 50S, components of the ribosome. The complete ribosome complex, along with initiation factors, mRNA and amino acids, is the complex for translation of the genetic code into a specific protein. Small, interfering RNA, siRNA, ranges from 18 to 25 nucleotides long. siRNA can bind to mRNA and prevent translation or enhance mRNA degradation. Other types, mRNA, may bind to single-strand DNA and thus prevent transcription, formation of mRNA. As a group, such RNA represent a specific and subtle element of control over protein synthesis. siRNA is an active area of drug development aimed at silencing or controlling genes. One of the main hurdles in developing this area of potential new drugs is delivery. Given that RNase, the ubiquitous enzyme for hydrolyzing RNA, is everywhere, how does one deliver such a labile molecule and give it enough persistence in the systemic circulation to reach its organ of activity or target tissue? In addition, siRNA may induce off-target effects or unwanted
Biotechnology capitalizes on the attributes of cells. In spite of the extraordinary diversity of cell types in living things, what is most striking is their remarkable similarity. This unity of life at the cellular level provides the foundation for biotechnology. All cells have the same basic design, are made of the same construction materials, and operate using essentially the same processes. DNA, the genetic material of almost all living things, directs cell construction and operation, while proteins do all the work. Now that we've familiarized ourselves with the structure of a cell, let's peek inside the nucleus and look at the structure of DNA. The chromosomes in the nucleus are highly coiled and condensed packages of DNA. When you zoom in on DNA, you can see that the DNA is arranged into functional units called genes. The Human Genome Project discovered that humans have approximately 20,000 genes on chromosomes. These genes are made up of individual DNA units called nucleotides, arranged in a specific sequence unique to each gene. Chromosomes, genes, and nucleotides are all DNA. They are just different levels of organization for the DNA. Let me see if I can explain that better. To use an analogy, it's like a book. The book is divided into functional units called chapters, and the chapters are made up of individual words. Chromosomes are divided into functional units called genes, which are made up of individual nucleotides. It was not until the mid-1900s that DNA was elucidated as the inherited material described in Gregor Mendel's P studies. Many scientists contributed to the current DNA knowledge, but most notably, Rosalind Franklin used X-ray crystallography to generate beautiful pictures of the DNA molecule. Watson and Crick studied these pictures, and along with all of the data that had been collected from previous experiments, published a paper in 1953 describing the structure of the DNA molecule. Let's look at the structure that Watson and Crick described. Remember that a nucleotide is the smallest unit of a DNA molecule. There are three components to a nucleotide. The deoxyribose sugar, shown in blue, is a 5-carbon ring. Although only the 5th carbon is shown, you can see that a carbon is represented by the meeting of 2 lines on the pentose ring. The carbons are numbered clockwise, starting from the central oxygen. The phosphate group bonds to the 5th carbon. Also notice that on the 3rd carbon, there is a hydroxyl or OH group. We'll discuss this in just a minute. The nitrogen base, shown in orange, is on carbon number 1. There are 4 nitrogen bases in DNA. They are adenine, guanine, cytosine, and thymine. Those 4 nitrogen bases are actually divided into 2 different classes. The purines have double rings and are made up of adenine and guanine. The pyrimidines have a single ring and are made up of thymine and cytosine. DNA is a double-stranded molecule. Notice that adenine, A, forms bonds with thymine, T, and guanine, G, forms bonds with cytosine, C. The bonds that hold these 2 strands of DNA together are highlighted in light blue and are called hydrogen bonds. These hydrogen bonds give DNA its characteristic helical shape. Also, if you know the sequence of DNA on one strand of DNA, you can figure out the sequence of the other strand because A always pairs with T and G always pairs with C. Therefore, the 2 strands are called complementary. There is another kind of bond, the bond that holds the nucleotides together on the single strand of the DNA molecule. Remember when we looked at the phosphate on the 5th carbon and the OH on the 3rd carbon of a nucleotide? Well, look closely and you'll see how the phosphate group on one nucleotide has formed a bond with the OH group on the nucleotide above it. This is called a phosphodiester bond, and these bonds are highlighted in yellow. The alternating string of sugar and phosphate bonds form the backbone of the DNA strand. One more thing. Notice that on one strand of the DNA molecule, the phosphate, or 5' end, is on top, and the OH, or 3' end, is on the bottom. What do you notice about the complementary strands of DNA? That's right, it's the mirror image. So on the complementary strand, the OH, or 3' end, is up, and the phosphate, or 5' end, is down. Because of the opposite direction of the strands, they are called antiparallel. This is going to become very important when we study how DNA replicates. Now that you understand the structure of DNA, we'll examine how the DNA information is made into proteins. Let's begin by examining the structure of RNA. RNA nucleotides are very similar to DNA nucleotides. However, there are some very important differences. First, the 4 bases in RNA are adenine, guanine, cytosine, and no, not thymine, but uracil. The second difference is found on the number 2 carbon. Do you see the purple highlighted hydroxyl group? This extra OH makes it difficult for RNA to form hydrogen bonds with adjacent nucleotides. That's why RNA is almost always single-stranded. And because it is single-stranded and represents only small sections of the DNA, RNA can leave the nucleus through small nuclear pores and travel into the cytoplasm, where it is used as a template to make proteins. There are 3 types of RNA to be discussed. Let's define them. Messenger RNA, or mRNA, is shown in purple, and is the copy of RNA that is made directly from the DNA sequence. The next 2 RNAs are necessary for making protein. Ribosomal RNA, or rRNA, is shown in brown, and along with proteins is what makes up the ribosomes. Do you remember what happens at the ribosomes? That's right, ribosomes are where proteins are made. The last type of RNA, called transfer RNA or tRNA, is shown in green, and it functions to bring amino acids to the ribosomes for protein assembly. The manipulation of genetic information, specifically DNA and RNA, is at the center of most biotechnology research and development. Modifications can be as simple as changing a single nitrogen base, G, A, T, or C, in a gene sequence, or as complicated as cutting out entire genes or gene sections and inserting new ones. Changing DNA sequences may affect the characteristics of cells or whole organisms.
5.2 Enzymes, Enzymology, Coenzymes, and Enzyme Kinetics. Enzymes are the executors of the cell. Enzymes are proteins that can catalyze chemical reactions. Enzymes are the machinery of metabolism. Enzymes are responsible for DNA synthesis and replicatio. They transcribe DNA into RNA, and they are essential to the process of translating the message of RNA into proteins. Enzymes are also responsible for protein breakdown and for the conversion of substrate, carbohydrates and fatty acids, into cellular energy. They are responsible for the synthesis of hormones and hence for cell-cell communication. They are indeed the machinery that makes cells work, because they are also responsible for muscle contraction and, hence, the real work of an organism. Enzymology. How exactly do enzymes function? They are basically catalysts. They assist in chemical reactions but are not consumed in those reactions. They accelerate the chemistry of getting from substrate to product. They accomplish this, as do all catalysts, by lowering the activation energy, the energy barrier to get from substrate to product. One of the characteristics of the chemistry of living things is that the chemistry must be done under relatively mild chemical conditions. Thus, most enzyme reactions must occur at reasonable temperatures, usually at 1 atmosphere of pressure, at relatively normal ionic concentrations, osmolarity, at reasonable ranges of phase, no harsh acid or base, and under a wide range of substrate and product concentrations. In addition, enzymes are very specific for the substrate work on and, hence, very specific in the products help produce. To accomplish these goals, enzymes work in a unique way. General catalysts, think of a platinum surface, are just that. They assist in a wide variety of reactions, such as breaking down a large hydrocarbon molecule into manageable pieces, i.e., octanes. However, enzymes are very specific and have an optimal substrate and favored product. Although some enzymes, i.e., cytochromes, will work on a small number of substrates with very similar chemical makeup, many enzymes will work optimally on only a single substrate and produce only a single product. In the instances when an enzyme will work on 2 or more closely related chemicals, one reaction will usually be highly favored and the other reactions will occur at much slower rates. The key elements of enzyme-assisted reactions are specificity, only one or a limited number of closely related substrates, and acceleration, reaction occurs manifold over what would normally occur. How do enzymes accomplish these twin goals? Many explanations exist, but they fall into the following basic categories. Asteric or induced fit. The active site of the enzyme is spatially configured to accept only one, or a very limited number, of substrate configurations. This model involves molecular size, orientation of reactive groups, ionic and hydrogen bonding, and possibly movement of the enzyme protein backbone or side chain orientation, fit, during substrate binding, catalysis, or product release. This model has replaced the original lock and key model of enzyme activity. Biolocalized chemistry. It has been suggested that the active site affords an environment for unique acceleration of the specific chemical reaction. As an example, the close, relatively asolvated interior hydrophobic core of an enzyme's active site might provide for unique acid-base chemistry. See transition state. The enzyme is able to form an intermediate with the substrate and, thus, lowers the energy barrier to the reaction of substrate going to product. In addition, the enzyme is able to break down a relatively complicated chemical change into relatively simple sequential steps, remove a hydrogen here, add a hydroxyl there, and so on. The proteolytic enzymes, trypsin and chymotrypsi, of normal digestion are good examples. They catalyze the hydrolysis of specific peptide bonds in the proteins of the diet. The substrates, a water molecule and a peptide bond, are transformed into a free carboxylic acid group and a free amine group, i.e., two peptides. A reaction that normally would require 24 hours of strong acid at 110 degrees Celsius has been accomplished in a microfractio of a second at physiological phase. The rapid rate of the peptide bond hydrolysis, acceleration of the reaction, and the limitation to a small number of peptide bond types, specificity of the reaction, are the two hallmarks of enzyme catalysis. This miracle of catalytic chemistry takes place every day in the digestive tract. Coenzymes. Coenzymes are small molecules that interact intimately with an enzyme and are required for the enzyme to express its catalytic activity. Coenzymes most often are associated with the active site of an enzyme or contribute to the chemistry of the enzyme-catalyzed reaction. In some instances, because the coenzyme binds to and then departs the enzyme after the catalyzed reaction, cofactors may appear to be more like substrates. In many cases, the coenzyme is covalently bound to the enzyme and functions in the chemistry at or near the active site. In a few cases, the actual role of the coenzyme seems to be intermediary between these two examples. An example here is adenosine triphosphate, ATP, which behaves like a substrate in most cases but can act as a modulator of enzyme activity in some cases. Several coenzymes are vitamins or are derived from vitamins. A good example is niacin, pyridine-3 carboxylic acid. This vitamin can be converted into two key coenzymes, NAD and ADP. Each of these has a hydrogenated product, AD and ADF, respectively, and the respective ratios, NAD to AD and ADP to ADF, are critical indicators or mediators of cellular energy production and oxidative reductive capacity because of their role as coenzymes. Similarly, flavin nucleotide and flavin adinucleotide are the two coenzyme forms of the vitamin riboflavin. Another form of coenzymes, although more appropriately labeled as cofactors, are the metals. Metals can be part of the active site or simply a stabilizing element of the enzyme tertiary structure. Metal ions as an element of the active site increase the type of chemistry available to facilitate reactions. After all, the side chains of amino acids and the chemistry of vitamins can provide only so much punch. Metals, acting as Lewis acids, for example, can provide an additional form of enzymatic catalysis. The two major groups of coenzymes are thus vitamins and metals. The most common metals associated with enzymes as cofactors are zinc, copper, magnesium, manganese, and iron. Table 5-5 lists the important water-soluble and fat-soluble vitamins and disease states associated with a deficiency in that vitamin. Table 5-5 also lists good food sources for that vitamin. Given the amount of processed foods in the usual Western diet, taking a daily multivitamin capsule is a reasonable recommendation. An enzyme without its cofactor is called an apoenzyme or apoprotein. An apoenzyme with its cofactor or cofactors is called a holoenzyme.
Today we're going to talk about cofactors and coenzymes, and how sometimes they can be essential to proper enzymatic function. But first, let's review the idea that enzymes make reactions go faster. And they do this by lowering the activation energy peak of their respective reactions. Let's also review the idea that enzymes bind their substrates at a location on the enzyme called the active site, which is where most of the reaction takes place. Now, not all enzymes are able to catalyze reactions on their own, and some need a little help. So if we have our enzyme here trying to react with our substrate over here, sometimes something called a cofactor or a coenzyme will be needed, which will also need to bind to the enzyme in order for it to function properly. And we're going to go over what coenzymes and cofactors are and exactly how they work. So first we'll talk about what a coenzyme is. Well, coenzymes are organic carrier molecules. And what I mean by organic is that they're primarily carbon-based molecules. And by carrier, I mean that coenzymes hold on to certain things for an enzyme to make the catalysis run a little more smoothly. And a great example of a coenzyme is NADH, which acts as an electron carrier. And here I've shown NADH dissociating into its oxidized form, NAD+, as well as to a hydride ion, which basically just exists as a pair of electrons that some other molecule would be grabbing. So NAD+ can accept electrons, causing the molecule to be converted to NADH, which could then carry electrons for an enzyme. Now, if you remember the lactic acid fermentation reaction, where pyruvate is converted to lactic acid, you'd see that the enzyme catalyzing this reaction, lactate dehydrogenase, uses NADH as a coenzyme in order to transfer electrons to the pyruvate molecule in order to turn it into lactic acid. And in this sense, NADH is acting as an electron-carrying coenzyme. Another example of a coenzyme is coenzyme A, which, like NADH, acts as a carrier molecule, but instead of carrying electrons like NADH does, coenzyme A, which we sometimes call CoA, holds on to acyl or acetyl groups instead. And you'd see CoA appear quite often in metabolic reactions, where it'll carry these two carbon acetyl groups from one molecule to another. Now, cofactors are a little different from coenzymes. While coenzymes are only really involved in transferring different things from one molecule to another, cofactors are directly involved in the enzyme's catalytic mechanism. They don't strictly carry something like a coenzyme would, but might be stabilizing the enzyme or the substrates, or helping the reaction convert substrates from one form to another. A great example of this is with the enzyme DNA polymerase. Remember that DNA polymerase is responsible for helping out with synthesizing new DNA during DNA replication. Now, you may remember that DNA is a very negatively charged molecule because of all the negatively charged phosphate groups that you'll find around it. Well, DNA polymerase uses a magnesium ion as a cofactor, which can use its big positive charge to stabilize all that negative charge on DNA. And you can see how this is different from a coenzyme, because instead of acting as a carrier molecule, the magnesium ion cofactor is stabilizing the DNA and is more directly involved in the actual catalysis. Now, interestingly, what people normally call vitamins and minerals, like the kinds that a doctor would tell you to make sure you get enough of in your diet, are often different cofactors and coenzymes. And what's special about vitamins and minerals is that your body can't build them up from scratch, and you need to get them from your diet in order to stay healthy. So when we say vitamins, we typically refer to organic cofactors and coenzymes. So two great examples are ones we just discussed. Vitamin B3, which you may see being called niacin on a food label, is actually just a precursor for NAD, and vitamin B5 is just a precursor for coenzyme A. Minerals, on the other hand, are inorganic, meaning they aren't carbon-based, and minerals are usually just cofactors in our body. So magnesium would be a great example of a mineral cofactor that an enzyme like DNA polymerase would use. Now, not all minerals act only as cofactors. Some minerals, like calcium, which can act as a cofactor, is also a critically important component of bone and teeth. And it doesn't strictly act as an enzyme cofactor here. It's actually an important part of the structure itself. So what did we learn? Well, first we learned that not all enzymes are able to function alone, and some need a little help. And next we learned that this help can come from coenzymes, which usually act as carrier molecules, or cofactors, which directly assist with the catalysis that the enzyme is doing. And finally, we learned that vitamins and minerals generally refer to dietary cofactors and So today we're going to talk about Michaelis-Menten kinetics in a steady state. But first, let's review the idea that enzymes make reactions go faster, and that we can divide the enzyme's catalysis into two steps. First, the binding of enzyme to substrate, and second, the formation of products. And each of these reactions has its own rate. Let's also review the idea that if we keep the concentration of enzyme constant, then at really high substrate concentrations, we'll hit the maximum speed for a reaction, which we call Vmax. So first, we'll talk about the steady state assumption and what that means. So like I said before, there are two steps to an enzyme's catalysis. Now, when we use the term steady state, what we mean is that we're at a point where the concentration of ES, or enzyme-substrate complex, is constant, which means that the formation of ES is equal to the loss or dissociation of ES. Now, notice that I've used equilibrium arrows between these steps, and that was to show the idea that these reactions, like any reaction, can go forwards or backwards. Our enzyme-substrate complex doesn't have to form products. It could just as easily dissociate back to an enzyme and a substrate molecule. So I'll call these reverse reactions -1 and -2. If we look at that in terms of our rates, we can say that the rate of formation of ES would be the sum of rate 1 and rate -2, since both of these reactions lead to ES. And the rate of loss of ES is equal to the sum of rates -1 and 2, since both of these lead away from ES. Now also remember that products very rarely go back to reactants, since these reactions are usually thermodynamically stable. So rate -2 is going to be so small in comparison to rate 1 that we can really just cross it out, which means that we can swap out that second double-headed arrow for a single-headed arrow. So using this information, let's do some math. Now I'm going to be deriving a new equation. This can get a bit confusing, so don't worry if you have a little trouble with this. Just rewind the video and try watching it a couple more times if you need to. So I'll start out by drawing the same sequence I did before, with the 3 different reactions, and I'll also write out that steady state equation I mentioned before, where we have rates forming ES equal to rates taking away ES. Now the first thing I'll do is swap out those rate values for their rate constants times the reactants for those reactions. So rate 1 will be equal to K1 times E times S, and so on for the other two. Next, I'll introduce a new idea and say that the total amount of enzyme available, which we'll call ET or E total, is equal to the free enzyme E plus the enzyme bound to substrate, or ES. And using this equation, I'm going to rewrite the E on the left side of our equation as the total E minus the ES, which would be equal to the E we had there before. On the right side of the equation, I've just factored out the common term ES. Next, I'm just going to expand the left side of the equation, so take a moment to look at that. And now what I'm going to do is I'm going to divide both sides of the equation by K1. So K1 will disappear on our left side, and on our right side I've put K1 in with all the other rate constants. Now since all these rate constants are constant values, I'm going to combine them in this expression of K-1 plus K2 over K1 into a new term Km, which I'm going to talk a little bit more about later. So in this next line, I've done two things. First, I've thrown in that Km value that I just mentioned, but I've also added ES times S to both sides of the equations, and thus moved it from the left side to the right. In the next line, I've done two things. First, I switched the left sides and right sides of the equation, just to keep things clear, but I've also factored out the common term ES on our new left side. And then what I'm going to do is I'm going to divide both sides of the equation by Km plus S. So I can move that term to the right side. So I'll make some more room over here, and now what I'm going to do is remind you that the speed of our whole process, which I'll call VO, is equal to the rate of formation of our product, which we called rate 2 before, which is also equal to K2 times ES. So now using our equation over here, I'm going to multiply both sides of the equation by K2. Now here's where it gets really tricky. Remember that if we're at our max speed, so a reaction speed VO is equal to Vmax, which happens when our substrate concentration is really high, then our total enzyme concentration is going to be equal to ES, since all of our enzyme is saturated by substrate, and there won't be any free enzyme left. So K2 times ET instead of times ES would be equal to Vmax instead of being equal to VO, like you see at the top. So I'll make some room here and then sub in K2ES for VO and K2E total for Vmax, and then we finally get to our end equation, which is called the Michaelis-Menten equation, and is super important when we talk about enzyme kinetics. So let's take a few steps back and talk about the Michaelis constant. So first, I'll write out the Michaelis-Menten equation, and if you remember, we created this new term which I called Km, but we never really talked about what it meant. So let's get to that. Now, if you bear with me for a moment and pretend that Km is equal to our substrate concentration, then we can sub in that value into our Michaelis-Menten equation, which would put 2s on the bottom, the sum of s plus s, and then the s will cancel out, and we'll be left with Vmax over 2. And what this means is that Km, which we call the Michaelis constant, is defined as the concentration of substrate at which our reaction speed is half of the Vmax. So when VO is equal to 1/2 of Vmax. And if we look at that on a graph from before, you'd see that Km is a substrate concentration specific to our circumstances, where our rate is at half of its max. And the lower our Km, the better our enzyme is at working when substrate concentrations are small. And we can use this Km term to quantify an enzyme's ability to catalyze reactions, which we call catalytic efficiency. So I'll rewrite the Michaelis-Menten equation. Remember, we defined Km as a substrate concentration, where VO is 1/2 Vmax. And since it's a concentration, it will be in units of molar, or moles per liter. But now I'm going to throw a new term at you called Kcat, which is equal to the maximum speed of a reaction divided by the total enzyme available. And we call this the enzyme's turnover number. And all this term is how many substrates an enzyme can turn into product in 1 second at its maximum speed. And we measure it in units of seconds to this -1, or per second, as in reactions per second. So we can define an enzyme's catalytic efficiency as a combination of Km and Kcat. And we do this by saying it's equal to Kcat over Km. So a higher Kcat or a lower Km would result in an increase in an enzyme's catalytic efficiency. And every different enzyme has a different catalytic efficiency in certain conditions. And we can use this term to score enzymes on how good they are. So we covered a lot of content in this video, but the really crucial points to remember are first, the idea of the steady state assumption that we make when looking at enzyme kinetics. And this is where we assume that the ES concentration is constant, meaning that the formation and loss of ES are equal. Second, we derived the critically important Michaelis-Menten equation, which you should consider committing to memory. And third, we talked about how you can score how good an enzyme is at speeding up reactions by looking at that enzyme's catalytic efficiency, which is a combination of two new terms we learned about, Kcat and Km.
Okay, Vmax and Km. Well, we need to be looking at graphs here. So this is the graph that's going to keep popping up all the time. On the y-axis is the rate of reaction, sometimes called V, and on the x-axis is the concentration of substrate, which is the purple things. And the little Pac-Men are enzymes. The IB wants you to know about 3 parts of this initial graph. So let's imagine if I have a concentration of 4 for the substrate, and 2 of them make contact and react with the enzyme. So I've got that dot on my graph. Let's try again with a concentration of 8 of the substrate. So this is a relatively low concentration. And in this case, 4 of them are reacting. So that's analogous to a rate of reaction of 4. Put those back, and still at this relatively low concentration, let's try 12 of them. And by looking at the graph, that's going to give me a reaction rate of 6, which is analogous to 6 Pac-Men eating 6 little wedges. Now this is the first of the 3 parts of the graph you need to know about. That's a linear relationship. So at low concentration, the relationship between the concentration of substrate and the reaction rate is linear, because there's lots and lots of enzymes available to do as much reaction as you need. So it's a straightforward linear relationship between the concentration of the substrate and the rate of reaction. There's more enzymes than you could use. Let's move up to 16 now. Now it's starting to get a little crowded there. There's not enough enzyme to accept every substrate that happens to find itself in the right position. So the rate of reaction is going to slow down a little bit there. So it's going to start to slow down in this curve at the top left-hand corner there, which is the second part of the graph you need to know about. The rate of reaction slowing down since the enzyme is starting to be, they're starting to fill up here. The substrate can't always find availability in the enzyme. Let's go crazy. Let's go up to 32. Well, now this enzyme, almost all of the enzyme, if not all of the enzyme, is being constantly used all the time. The maximum rate has been reached. The enzyme is full. And even if I was to double the amount of substrate up to 64, that's not going to change the rate. The enzyme's always busy, always being used with the substrate. And so that's exactly 10. Oh, not quite. Let me just fiddle. It's exactly a rate of reaction of 10. Don't forget the units are probably going to be molar per second or something. And that's called Vmax. So Vmax is 10 for this. Now, if I was to divide Vmax by 2, giving me 5, and read down to the concentration of substrate, that gives me Km. Capital K, small m. Units of concentration there, because it's concentration of substrate. And that is the Michaelis constant. Mr. Menton's probably upset that he doesn't get a mention in the IB syllabus. And again, that's at Vmax over 2. So let's draw this graph out again. V, don't forget, it's velocity, which is the rate of reaction. And here are two enzymes, enzyme X and enzyme Y. Now you can read across and see that they have different values for Vmax, maximum velocity or maximum rate. So we'll discuss what that means in a second. And also they have different values for the Michaelis constant. So if I divide Vmax by 2, that gives me the Michaelis constant for Y. And for X, if I divide Vmax by 2, that also gives me the Michaelis constant for X. And don't forget, V is rate or velocity, if you will. So for Y, you can tell that that's going to be faster because it has a higher Vmax. And X is going to be slower. It has a lower Vmax. And having a lower Km, well, that means the reaction is going to be faster at low substrate concentrations. And so Y is going to be faster at low substrate concentrations. And a higher Km, well, you can interpret that as it being less reactive. The enzyme-substrate reaction is less reactive. And that gives you a higher value of Km. So let's look at Km once more. So for a specific enzyme, there are three things that can change Km, the Michaelis constant. And they are temperature, pH, and the presence of heavy metals. Now, don't think you're done with these graphs. More graphs are coming up a bit later with the competitive and non-competitive inhibitors.
Let's see how we can make a Lineweaver-Burk plot, a double reciprocal, starting with the hyperbolic plot of the Michaelis-Menten equation. First, we'll take the reciprocal of the velocity of the reaction. Next, we take the reciprocal of substrate concentration. Next, let's plot the points. The result is a linear plot, the Lineweaver-Burk plot. We can determine the Michaelis constant and the maximum velocity more easily from a linear plot than a hyperbolic one.
ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
And please post some interesting things, such as your artworks. And you can also please change the settings to original or HD so you can have better graphics to see the videos. To see this video, which is on fatty acid degradation, for example. Yes. So in the previous video, we learned how fatty acids were transported from the cytosol, where it was synthesized, to the mitochondria. Now, in the mitochondria, fatty acids can undergo fatty acid oxidation when we need energy from fatty acids in the form of acetyl-CoA. Fatty acid degradation, or full oxidation, comes in 3 stages: stage 1, stage 2, and stage 3. Stage 1 is when a long fatty acid chain, a long fatty acyl-CoA, such as palmitoyl-CoA, a 16-carbon fatty acid, can undergo beta oxidation, which will essentially cleave two carbons at a time from the long chain fatty acid. So in this case, palmitoyl-CoA, which is a 16-carbon, through beta oxidation will make 8 acetyl-CoAs. Okay, now during the beta oxidation process, electron carriers will be produced, such as NADH and FADH2. Let's leave it there for a second. Now let's go back to the 8 acetyl-CoAs. Now the 8 acetyl-CoAs can then go on to the next stage, stage 2, by entering the Krebs cycle. Because if you remember, acetyl-CoA enters the Krebs cycle from pyruvate. Same thing here. And from the Krebs cycle, NADH and FADH2 are also produced, which leads to the final stage, stage 3. So in stage 3 is about the electron carriers. NADH and FADH2 gets fed into the electron transport chain to produce more ATP from ADP and inorganic phosphate here. So to produce more ATP. Just like cellular respiration. But what this video is actually more concentrating on is on stage 1, which is beta oxidation. So let's have a closer look. So beta oxidation, what is it? And why beta? Why not alpha, for example? Well, if we take a single fatty acyl-CoA, such as palmitoyl-CoA, which is a 16-carbon fatty acyl-CoA, degradation of fatty acids begin at the carboxyl end. So here is a carboxyl end here. Now, the nomenclature of fatty acids using Greek alphabets begin with the first carbon, which does not include the carboxyl carbon. So here, for example, is alpha, beta, gamma, and delta. So beta, beta. In beta oxidation, we want to oxidize beta. We want to add oxygen to beta carbon, this beta carbon here. So through several reactions, this same palmitoyl-CoA structure will look like this. And here is a carboxyl end still. And what is different is that the beta carbon is now bonded to oxygen. So it has been oxidized. Nice, that's a good spelling of oxygen. So next, if we add a CoA group to the structure, we can have the beta carbon bind to the CoA group, which will release the alpha carbon and the carboxyl end. And this is acyl-CoA, what's split out. So, but seeing we release 2 carbons, it means that this structure is not a 16-carbon fatty acyl-CoA anymore. It's a 14-carbon fatty acyl, which also means that this is no longer alpha, this is no longer beta, gamma, and delta. Here is the new carboxyl end, which means here is a new alpha carbon and beta carbon. And so beta oxidation can continue on with this 14 carbon. So from 16 carbon fatty acid from the very beginning, if we remove 2 carbons at a time, we will get 8 acyl-CoAs. So let's go back to the map. We will start with palmitoyl-CoA. Just a point to make, fatty acid degradation is the reverse of synthesis. So if you remember, synthesis was condensation, reduction, dehydration, and reduction again. Well, if we go backwards, it's oxidation, hydration, oxidation, and finally cleavage. Okay, so palmitoyl-CoA, it will oxidize first to trans-2-enol-CoA by the enzyme acyl-CoA dehydrogenase. FAD is reduced to FADH2. Next, trans-2-enol-CoA will then be hydrated to L-beta-hydroxyacyl-CoA by the enzyme enol-CoA hydratase, because you're adding water to it. Now, L-beta-hydroxyacyl-CoA will then be oxidized to beta-ketoacyl-CoA. Beta-ketoacyl-CoA by the enzyme beta-hydroxyacyl-CoA dehydrogenase. NAD, beta-hydroxyacyl-CoA dehydrogenase. Now, NAD is reduced to NADH. So, from the beginning, we had oxidation, hydration, and oxidation again, which means that finally, beta-ketoacyl-CoA will cleave 2 carbons off, forming 14-carbon fatty acyl-CoA called myristoyl-CoA. The CoA was obtained in the reaction. And also, the second carbon, also the 2 carbons with CoA that broke off, is acyl-CoA. It forms acyl-CoA. So that was how we formed one acyl-CoA. But myristoyl-CoA can still undergo beta oxidation 6 more times to form more acyl-CoAs. So as we have learned, fatty acid degradation is the reverse of fatty acid synthesis. So whereas fatty acid synthesis is condensation, reduction, dehydration, and reduction, fatty acid degradation is backwards of this. So oxidation, hydration, oxidation, and cleavage. I hope that makes sense. I hope that was good. Next, we will look at acetone, how ketone bodies are formed, and how ketone bodies serve as important fuel reserves in times of starvation. Thank you. Please like, comment, and subscribe. Thank you all.
COA. Acetoacetate, 4 degrees Celsius, beta-hydroxybutyrate, 4 degrees Celsius, and acetone, from acetoacetate, are commonly listed as keto bodies. Their concentrations can rise in ketosis or ketoacidosis in patients with diabetes mellitus. This condition can be life-threatening. However, the brain and heart use keto bodies as a direct energy source through reconversion to acetyl-CoA. Acetyl-CoA plays a central role in energy production from amino acids, fatty acids, and carbohydrates. Acetyl-CoA can then be used to synthesize sterols, fatty acids, and keto bodies, or can be used in the tricarboxylic acid cycle to produce additional ATP. 5-4 Nucleic acid metabolism, DNA replication and repair, RNA transcription, and translation into protein. A review of nucleic acid metabolism must begin with an understanding of the nucleus. The nucleus is the largest intracellular compartment and is surrounded by 2 membranes called the nuclear envelope. Within the nucleus is a subcompartment called the nucleolus. DNA is contained within the nucleus as a highly organized and tightly coiled protein or DNA containing complex called chromatin. See Chapter 6 for more details. Bare DNA does not normally occur. The chromatin is further organized into chromosomes, which vary in number depending on the species. The protein complex with DNA serves to compact the lengthy DNA molecules into a tighter, denser form and to add an element of access control to DNA for translation and of regulation of that translation. The main biochemical reactions in the nucleus include replication of DNA during mitosis, repair of DNA following damage, Table 5-6, transcription of DNA into RNA, and processing of that RNA into a specific mRNA. Part of the processing of RNA occurs in the nucleolus. Translation of that message into protein synthesis, occur in the cytoplasm. See Chapter 6 for additional descriptions on transcription and translation. 5-5 Recombinant DNA technology. Synthesis of proteins takes place in the cytoplasm on a complex structure called a ribosome. The ribosome consists of 2 large lobes, termed 305 and SOS, formed of multiple proteins associated with ribosomal nucleic acids specific to each lobe. Ribosomes are usually attached to the rough reticulum. Synthesis also requires an mRNA, which specifies the sequence of amino acids. A supply of charged tRNAs, the various amino acyl tRNAs, is also required. Figure 5-10 depicts the transcription of DNA into an mRNA in the nucleus, the movement of the mRNA into the cytoplasm, and the translation of the mRNA sequence into a specific protein. Note the alignment of codons in the mRNA with the corresponding anticodo at the bottom of each tRNA. See Chapter 6 for more specific information on mRNA and tRNAs, codons and anticodos. Thus, the specific sequence coded in the DNA is transcribed and then translated into a specific sequence of amino acids, a protein. The process is more complicated than shown in Figure 5-10 and requires initiation factors, elongation factors, energy, and a specific stop codon to complete the process of synthesizing one protein chain. In vivo, many ribosomes act on the mRNA at the same time, but at varying points along the message. Thus, many protein molecules are being synthesized at the same time and are in various phases from initiation to completion. In recombinant DNA technology, this process is adapted to overproduce a specific protein of interest, a microorganism, bacteria or yeast, or a cultured cell line. The process starts by identifying the DNA of interest and then cloning that DNA sequence into the native DNA of the microorganism or cultured cell line. Using factors to promote or enhance the expression of the specific protein of interest, one can manipulate the system to overexpress the needed protein. Isolation of the protein of interest may be from the culture media, secreted protein, or may require cell lysis and purification of the protein of interest. Obtaining a protein with the proper post-translational modification or glycosylation to match the original mammalian protein, from the cell culture or microorganism culture, can be a challenge. Such recombinant therapeutic proteins are expensive to produce and available in only limited quantities. Monoclonal antibodies produced by recombinant technology are one of the largest areas of new therapeutics currently in the clinical trials arena and will continue to be an active area of new drug development for the foreseeable future. 5 to 6 key points. At the cellular level, a highly complex, redundant, and highly regulated macromolecular and molecular set of machinery maintains the negative entropy that is known as life. At the tissue and organism level, this level of regulation includes internal, external, and cross-species forms of communication and regulation, at least in the gastrointestinal tract. Enzymes are proteins that have a primary role as catalysts for the myriad reactions necessary to carry out cellular development, cell metabolism, growth and differentiation, and cellular regulation, and they ultimately control cell death, apoptosis. Amino acids are the building blocks of proteins. See Table 5 to 1 for a listing of amino acids. Some protein amino acids are key elements of metabolic pathways. Citrulline and ornithine in the urea cycle are good examples. Protein and enzyme structures have four main elements: primary, linear sequence of amino acid; secondary, alpha helices and beta pleated sheets; tertiary, proper folding of secondary structures into native state; and quaternary, combination of two or more protein chains to form a multi-subunit protein complex. SNP, single nucleotide polymorphism, or mutational change in a DNA coding sequence, can lead to an alteration in the primary amino acid sequence of the resulting protein. This SNP can result in a silent mutation, not clinically observable, or a change that results in a clinically significant syndrome such as sickle cell anemia. Carbohydrates are a major source of energy. In humans, carbohydrate energy is stored as glucose predominantly in the liver and muscle. Fatty acids are a major source of energy but also are critical components of cellular membranes as phospholipids. The nucleotide units of both DNA, at the cellular level, a highly complex, redundant, and highly regulated macromolecular and molecular set of machinery maintains the negative entropy that is known as life. At the tissue and organism level, this level of regulation includes internal, external, and cross-species forms of communication and regulation, at least in the gastrointestinal tract. Most vitamins act as coenzymes and are small molecules that interact intimately with an enzyme and are required for the enzyme to exert its catalytic activity. Certain metals can also act as coenzymes. The Michaelis-Mete equation, V equals V max, /1 plus kilometer /S, equals Fmax, S, kilometer plus, S, is used to mathematically describe enzyme activity. Plotting 1/V, inverted velocity, on y-axis versus 1/S, inverted substrate concentration, on the x-axis is one way to determine kilometer and V maximum. The main biochemical reactions in the nucleus include replication of DNA during mitosis, repair of DNA following damage, transcription of DNA into RNA, and processing of that RNA into a specific mRNA. Part of the processing of RNA occurs in the nucleolus. Monoclonal antibodies produced by recombinant C-gluconeogenesis in liver technology are one of the largest areas of new muscle and liver glycolysis therapeutics currently in development.